In this video, I'm going to run you through some of the more important concepts in Chapter 9, which is about microbial growth. First thing we're going to talk about in this chapter is generation time. Generation time is how long it takes for a particular species of bacteria to double or to reproduce itself. Every species is different, and it's different depending on the environment that it's growing in. For example, the generation time would be different if it's growing in a refrigerator versus if it's growing at room temperature. Generally speaking, generation time would be something that's going to be given to you. Here they're telling you, like, this particular species on a specific medium, how fast is it able to double? And this is how things are going to tend to be given to you, is we're going to tell you the species and then how fast is it able to double. I'm going to tend to make the numbers nice round numbers to make the math a little bit more simple to understand. The formula for calculating generation time. So these two things are essentially the same, and N is the way that the Openstax book gives it, but NT is usually how it's represented, where that's the population at a specific period of time that you are trying to calculate. N sub O is the number of bacteria or the number of organisms that the population started with, and 2 to the nth power, where N is the generation time, or the number of times that we could divide given the amount of time that the problem has given us. So we're going to go over an example problem that I have given you here. So if a culture medium is inoculated with a cell that has a generation time of 20 minutes, how many cells would be present in the culture after 5 hours? Okay, in order for you to set this problem up, you've got to remember the formula that was given to you just a second ago. And you've got to remember, I got to find my correct pin, probably should have had that out a second ago, but you're trying to figure out how many organisms are present after 5 hours. Okay, that's going to equal, we started with 1 cell. So 1 cell times 2 to the nth power. This is the hardest part for most of my students to be able to figure out. That N, that superscript N, is how many times the organism was able to divide given the period of time allotted in the problem. So the generation time is 20 minutes. The period of time given is 5 hours. So how many times can 20 minutes go into 5 hours is what you have to figure out here. And here's where you really have to be able to remember an analog clock and how many times 20 minutes goes into an hour. So there's 60 minutes in an hour. 20 can go into 60 3 times. That means it can go into 5 hours. Well, if it's 3 for 1 hour, that means 5 hours is 15 times. So this N is going to become 15. So 2 to the 15th power is what that's going to be. So written nice and neatly. That's the same thing that I have just written out. From there, it becomes a matter of plugging that into your calculator. Every calculator is a little bit different here, but once you have it set to the scientific mode, you're going to do 2 to the 15th power and then equal, and then times, in this case, the 1, just to make sure you're practicing that you always have to do that original population that you started with, and then that's going to give you your final value. And so in this case, that's 32,768 cells. So after 5 hours, we had started with 1 cell. After 5 hours, we have over 32,000 cells present in that population time. And that shows you, 1, how fast bacteria are able to grow or double in population size. And 2, how you're able to calculate that. I've given you another example problem, and the PowerPoint shows you how to do that. And so if you need some practice with that, do that. And the book also gives you some practice problems. If you're struggling with that, please don't hesitate to contact me so that we can Zoom and have a meeting about it or email through it. Next thing your book talks about is the growth curve. Growth curve is what you see when you're growing organisms in batches, like on a slant, in a broth, on a plate, whatever, essentially. So you start out by putting your organism into that batch. And at first, the organism's going to kind of be stunned. You just put them on there. You're going to have this lag phase where they're not increasing in size. And by size here, I'm talking about the population, not the size of the organism or the cell, but the size of the population as a whole. Then after that little period of adjustment, we're going to start doubling the population with the generation time. So for E. coli, it's approximately every 20 minutes. For Mycobacterium, it's approximately every 12 hours. Every species has a different generation time, but we're going to enter what's known as the log phase or the exponential growth phase, and that's where the population is doubling every generation time. After that, what happens though is the nutrients in the medium that we were growing in are consumed. We're running out of those nutrients. The waste products, they're accumulating. We're essentially being poisoned by our own waste. And so the number of organisms dying starts to equal the number of organisms that are being produced. And we enter the stationary phase where, yeah, we're still producing new organisms, but we're producing them at the same rate that they're dying off because of essentially the poisons that are being created by those same organisms. After that, the nutrients are depleted so much that we start to starve to death, and then we enter the phase of prolonged decline or the death or decline phase. Essentially, we're never going to more than likely kill every single thing in there. What starts to happen is essentially evolution in this phase, where different strains within that population start to do better while other strains start to struggle. The population as a whole is going to decline, but certain cells are going to do better than other cells during that phase of prolonged decline. Okay, culture density, there's a whole slew of different ways that you can measure that. It's one of those things that's very advantageous to know. One of the sayings in life or medicine is the dose makes the poison. If you drink a cup of water that has 1 E. coli cell in it, you're not going to get sick from E. coli. You have an immune system. It's going to be able to deal with that. But if you drink a cup of water that has 8 billion E. coli cells, you're definitely going to get sick from that. That's a different culture density between those two things. 1 cell per cup versus 8 billion cells per cup. So culture density is the number of cells per unit volume. There's a whole bunch of different units you can use, but one of the more common ones is cells per milliliter that you can see right there. This is just what's happening during those phases. I really already gave you that. The importance of the log phase is a whole slew of different things. One's generation time does get calculated during that phase. 2, primary metabolites are being made during that phase. Antibiotics work best during that phase. Disinfectants work best during that phase. It's medically relevant, that phase. And so you want to try to treat infections during that phase. If a person waits a long time and then they seek treatment, they're not in the log phase anymore. The bacteria have already hunkered down. It gets more difficult to treat that infection because we're already doing that evolutionary arms race inside of that person, and so medical treatment becomes more difficult. So we want to try to get that treatment started very early. It is possible to do not a batch culture, if that makes sense. We can maintain a log phase in a lab setting. When we do that, essentially what we're going to do is we're going to pump out organisms at about the same rate that organisms are produced. If we're trying to mass produce organisms because the organisms are producing economically viable products for us, like vaccines or medicines or just whatever, we want to maintain them in the log phase so that they can produce those products for us. And so chemostats are a way for us to do that. It's just a way that we do that in a lab setting, not really a medicine thing. This is more of a commercial pharmaceutical type thing. Okay. From here, your book switches to what do organisms need in order to survive? Now, you've been through A&P or freshman biology, and you know what humans need to survive. You know, you need oxygen, you need nutrients, you need water, you need proper atmospheric pressure, you need a proper temperature. Well, organisms, microorganisms need that too, but their requirements are vastly different from yours. There are organisms out there that actually get poisoned by oxygen, and if they are in the presence of oxygen, they will die. There are organisms out there that prefer a high carbon dioxide environment and a low oxygen environment. There are organisms out there that prefer to live in a crazy acidic environment that would kill you if you were to set foot in it. So that's what your book is sort of talking about here, is every organism has its own set of requirements that it needs, and if you take it out of those requirements, it will either die, or it will not grow as well, or things will happen to it that will not be ideal for that. And so it starts to just give you terms that those organisms, that we apply to those organisms to sort of describe them. Capnophiles, halophiles, halotolerant, facultative anaerobes, all those sorts of things. Those are just terms for us to use to describe what organisms need in order to survive. The reason that this is very important for you is for you to understand that we can't grow all organisms in labs. We don't have all the media. We don't have the proper atmospheric pressure for some. We can't maintain the proper atmosphere. And by that, I mean gaseous mixture for some. We don't know the requirements for some. They're so picky about what they need. We just literally cannot grow them. And so we're still learning a lot about the requirements that a whole slew of different organisms need. And so that's an important concept. We know a lot about what the pathogens that prey on humans need, but we don't know a lot about the things that don't prey on humans need. Let's see, the next thing that I kind of wanted to talk about is for those things that we can grow in lab, there are a whole bunch of different media that we can use, but again, they really only work on certain organisms that are mesophiles. In other words, they grow best around room temperature. They tend to be either aerobic or facultative anaerobes because it gets hard to work with organisms that are anaerobic. You need specific things to work with them. They tend to need like moderate amounts of salt, not too salty. They tend to need a relatively moderate pH. Those are just the things that we learn to grow the best. I always like to talk about chocolate agar just briefly because students always assume it actually has chocolate in it, and it doesn't. It's just broken down red blood cells. So it's kind of like blood agar, only blood agar has full and intact red blood cells versus chocolate agar, the blood cells have been lysed or broken down. From there, honestly, most of the rest of this chapter gets covered in lab pretty well. What I really just want to mention is that a lot of the media that you use in lab can be classified into numerous different categories here. So just because something is selective doesn't mean it can't also be differential. Just because something is complex doesn't mean it can't also be selective. A lot of times one agar fits into multiple different categories. For example, mannitol salt agar is both selective and differential. And so just keep that in mind as you go through there, but this is really where lab is going to help you out. I hope that this video helped explain some of the more difficult concepts, but remember, as always, if you struggle with anything in this chapter, please contact me. I am here to help you.